Okay. So if we start zooming in on a neuron, and we'll focus on the cell body at first, because again, that's where all the basic cellular machinery exists. What we end up seeing is that there's a neuronal membrane around to the edge. This is just the cell membrane. Sometimes they give special names to things that are from a neuron. So sometimes you'll hear this called a neural lemma, but it's basically the outer cell membrane. And the cell membrane is not just an envelope that contains all the structures that exist within the cell, but it's also really fundamental to the function of how cells work. Cell membranes for neurons, like any other cell, are composed of phospholipid bilayers. This is a little picture I always liked. This is just like a little chunk out of the membrane. So this might be the outer surface of the membrane, and we've just kind of like carved out a little square of it and then zoomed in on it. And what you can see is that there are all these little phospholipids, which are these little red bead-like things. And they have two different polarities, or two different structures to them. Their head groups, the little bead-like structures up at the top, are polar. And so as a result, they are attracted to water, and they are hydrophilic. The same is true on the inside layer. We have another layer of these little head groups from these phospholipids. But each phospholipid molecule, you can see, has these little strings that are sticking off it. We call this a fatty acid tail. The fatty acid tails are hydrophobic. They are nonpolar. And so as a result, you get this layer within the cell membrane that is all hydrophobic and creates kind of a barrier that separates the inside and the outside of the cell. And it's by moving materials, particles, especially ions, across this membrane, typically not going directly through the membrane, but instead going through these green things, which are proteins, that we're actually able to cause charge differences across neurons. And those charge differences are fundamental to how the things actually behave. That's basically what we're going to be focusing on in chapter 3 and 4, our next two chapters after this one, is how exactly those membrane potentials, we call them, are generated and function to allow for the function of neurons. As I mentioned, these green things that are sticking off of here, these are proteins, and these things do all kinds of functional things for the neurons. So these could be receptors, or they could be channels that allow ions to flow from one side to the next, or they could be enzymes. They could be structural proteins that are helping to hold the neurons together and maintain its shape. And those are embedded within that membrane. Now, as we dive into the cell body, deep within the cell body would be the nucleus, and the nucleus is visible under Nissl stains. They tend to be fairly large. But this is, I think as probably everybody knows, this is the housing spot where we have all of the DNA that exists within this neuron. And these neurons, the neurons in your cells, will have the same DNA that's found in all the other cells that you have, except for your gonads. Or your gametes at least. And so the neurons' genetic information is the same as the other cells, but there are differences in terms of which genes are expressed in neurons versus other cells that give them their unique properties. All the DNA that's housed within the nucleus is contained within chromosomes, and the chromosomes typically are lightly wound in an active neuron, such that there are proteins that can then get access to the DNA within those chromosomes and make use of the DNA to produce proteins. So there's sort of a multi-step process, and this is referred to as the central dogma of molecular biology. The basic idea is that we start with DNA, and then the DNA is turned into messenger RNA. That didn't work. Which is then subsequently turned into protein. Each of these steps has a different name. So the first one that's listed up here is gene transcription. Transcription is this process of going from DNA, which is like our master template, master blueprint of all genetic information in the cell, and then we transcribe it. Transcribe just means copy. But transcription. So we transcribe the DNA, or at least a fragment of the DNA, into messenger RNA. Messenger RNA then is this working copy that we take out of the nucleus and use to actually create a protein. This step of going from messenger RNA into protein that exists, that occurs outside of the nucleus, is called translation. So the idea is that DNA is kind of just like RNA. There's a few slight differences in the molecules. And again, I think probably you've heard this story before. So we call that transcription. We're just copying from one nucleic acid language to another nucleic acid language. Whereas going from messenger RNA to protein, that's going from a nucleic acid language to an amino acid language. So it's a different kind of molecule. So we're kind of translating into a new language. So we call that translation. All the transcription that happens within the cell happens within the nucleus. And that's what they're trying to depict through these different images that are happening here. So you start with DNA, and so in this case what they're showing here is one gene, so one part of it, one chromosome. So this is, just a physical structure. There'll be some kind of promoter region that is early on in the DNA that allows for various machinery to bind, and then there's a specific enzyme, it's called RNA polymerase, that will bind here and then basically just march its way down the DNA molecule till it hits a terminator. And along the way, it'll produce a copy of RNA. In fact, that's pretty nicely depicted here. So here's your DNA. Here's transcription, where you're taking that DNA and producing with this RNA polymerase enzyme they're showing as a dot, you're producing this working copy of RNA. And that's what they're depicting here as well. So here's your DNA, you undergo transcription, you create a working copy of that little fragment in RNA. What ends up happening, though, is that only a part of that fragment of RNA that we've just produced through RNA polymerase is actually functional, or at least is actually going to be used to create the protein. Those areas that are actually going to be expressed are called exons. So X stands for, the X in exon stands for expressed. It's the part of the DNA that's actually going to be expressed. And so what we end up doing is we have to undergo this process called splicing, where from this messenger RNA transcript we just produced, we have to basically cut out all the non-coding regions, the parts of the RNA molecule that are not going to be used to create protein. And we kind of put them together into a mature messenger RNA transcript down here, and this is the thing that is actually going to leave the nucleus and be used to create a protein. All these other bits that are out here are not going to be used to actually encode a protein. They tend to be locations where, sometimes people just argue these are junk RNA regions or junk DNA regions. Some people argue these are like remnants of old viruses that have inserted their nucleic acids. Oftentimes, these actually do have function in that they'll be like on the DNA molecule. These might be sites where certain transcription regulators will bind to, so you get to help regulate which genes are being expressed through interaction with these other areas, these intron regions, which are kind of in between. But the important ones are the exons again, and they get spliced together to form this mature messenger RNA transcript. Once we produce a mature messenger RNA transcript, that's this little red thing here, it will then leave the nucleus via nuclear pores and go out into the cytoplasm of the neuron. And it's there that we can actually do this next step, which is translation. Okay, so the messenger RNA at this point now, it's exiting through nuclear pores. And what will happen out there is that we'll encounter a small structure that's actually composed of RNA itself called a ribosome. Ribosomes, in this case, in this picture, are drawn as these little dots, these little circular structures that are all interacting with this other thing that's called ref endoplasmic reticulum. Some ribosomes can just be sort of freely floating around, but oftentimes what will happen is that the ribosome will bind onto a messenger RNA transcript and then will dock with the ref endoplasmic reticulum, and then that's the site at which this step of translation will occur. So what will happen is, so here's your messenger RNA in this, again shown as this little red line. A ribosome binds to it, and then as the ribosome moves down the RNA, it uses the information that's in the RNA, the nucleic acid sequence there, to create a protein, and that's this little yellow thing that's being produced. And this will have a particular amino acid sequence that's based on the nucleic acid sequence. In some cases, like I mentioned, the RNA will just bind onto free ribosomes. And these will just be, like little dots out here that will be circulating out in the cytoplasm. In many cases, proteins are going to be destined to be part of some organelles, so they'll be embedded in a membrane elsewhere within the cell, or they might be destined to be part of the membrane on the outside of the cell, so the cellular membrane itself. Or maybe they're destined to actually be released. So for example, we do make peptide protein, peptide-based neurotransmitters that are produced in this manner, and then are going to be released from the cell. In all those cases, what we instead do is we have the endoplasmic, or the ribosome will dock with the endoplasmic reticulum, as we're seeing here. And then as the ribosome reads down the messenger RNA molecule, it produces this protein, but injects the protein either directly into the lumen of the endoplasmic reticulum, or it actually will embed it in the membrane oftentimes if it's going to be a membrane-bound protein that'll go on the cell surface. Like if you're making a receptor for a neurotransmitter, you'll directly embed that protein that you just manufactured in the membrane of the endoplasmic reticulum. And then from there, that protein and even that little snippet of membrane that it's embedded in will be transported over a series of steps to the cell membrane's surface. So we call this organelle, this membrane-bound organelle that we're seeing here, this thing is called the rough endoplasmic reticulum, or rough ER. You do need to know endoplasmic reticulum, so there it is spelled out. It's called rough because when you look at it under an electron microscope, you literally can see the little studs of all these little ribosomes sticking off of it. Because all the time there'll be ribosomes producing proteins at any given moment, and some of them will be attached to that endoplasmic reticulum. That is to distinguish it from other types of endoplasmic reticulum, which we'll see on the next slide, which is referred to as smooth endoplasmic reticulum, where you do not have that same sort of process where ribosomes are docking onto that structure. And there's other things that endoplasmic reticulum can do aside from being kind of like the landing spot for these newly synthesized proteins. The reason I'm going through all this is because what we'll see in the next several chapters is that most of the really important structures that are responsible for the function of how neurons work in terms of generating membrane potentials and interacting with one another, the receptors, the enzymes that are all associated with these things, these are all proteins. And so those proteins are all manufactured through the same basic process. So I think it's important that we understand exactly how that works, because at some point we may need to understand what goes wrong. I see a hand? No. Okay. So that's a rough endoplasmic reticulum. That's basically, in a nutshell, that's the first major steps of producing a protein. We've gone through all these major stages at this point. There are lots of other organelles that are present within neurons, though, and some of them are directly related to this process. Some of them are not really. So we do have this whole endomembrane system, which are these basically a collection of membranous sacs that exist within the cell, and many of them do not have ribosomes, unlike the rough endoplasmic reticulum. So, like I was just saying, there is smooth endoplasmic reticulum that exists in cells as well. I actually don't have a good picture of it on this slide. It might be on a subsequent one. Smooth endoplasmic reticulum can do a number of different things for neurons, though. So it actually can be a site of protein folding. So sometimes ribosomes will inject proteins into the rough endoplasmic reticulum, and then they can make their way into smooth endoplasmic reticulum because, and oftentimes, the smooth endoplasmic reticulum will just sort of be a branch off of the rough endoplasmic reticulum. If that's true, then proteins can be modified. So we can take those newly synthesized proteins that were shown on the prior slide as like little yellow squiggles, and we can fold them into complex shapes. And sometimes that will happen within smooth endoplasmic reticulum, as well as other structures that we'll talk about. Smooth endoplasmic reticulum is really important in terms of calcium regulation. This is especially critical in skeletal muscle cells, which we won't talk a ton about in this class. But skeletal muscle cells basically house all of their calcium within smooth endoplasmic reticulum. They call it sarcoplasmic reticulum, but it's smooth endoplasmic reticulum. And then when you stimulate a muscle cell, you'll have calcium be released from that smooth endoplasmic reticulum off into the interior of the cell, and that's what triggers muscle contractions to happen. So, and then ultimately you pump the calcium back into the smooth endoplasmic reticulum and store it until you again stimulate those same muscle cells. So smooth endoplasmic reticulum is critical for calcium regulation as well as some other ion regulations. And it's also critical for phospholipid synthesis. So phospholipids are those little molecules that we talked about that are fundamental to the cell membrane. They're also basically what composes the endoplasmic reticulum and the Golgi apparatus and the smooth ER. All the membranes in and outside of your cells are composed of phospholipids. And we make a lot of them in the smooth endoplasmic reticulum. So that's a major function for smooth ER as well. Another major membranous sac that exists within your cell body will be your Golgi apparatus, named after that same guy, Camillo Golgi, because he's the one that discovered the thing. Oftentimes this is referred to as like a shipping and receiving center. So the idea is that your ribosomes create and translate proteins and inject them into the endoplasmic reticulum. So that's this blue structure here, the rough ER. And then they basically get packaged in little tiny vesicles, which are just little tiny sacs of membrane. And here are the proteins that are embedded in there. And they'll be transported then to the Golgi apparatus. And the Golgi apparatus receives those little vesicles and basically incorporates the vesicles. And then gradually those proteins that were brought by the vesicle will make their way through the multiple sacs of the Golgi apparatus until it gets to this outer facing end. Do I have cis and trans on here? I don't think I do. That's okay. So basically this is the inward facing side of the Golgi apparatus that's receiving those those newly synthesized proteins. This is the outer facing layer of the Golgi apparatus, and it's basically going to then be in charge of distributing. So we're sort of receiving all the information over here, all the proteins over here. And then they make their way through this structure. And the structure, while they do so, there will be some modifications to those proteins. So little bits can get clipped off of them or cleaved off to them, or other bits can be added to them, or they can be refolded in different ways as they're making their way through the structure. But then ultimately there'll be some little tag, so basically some amino acid sequence that'll be present on each protein that you're manufacturing. The Golgi apparatus effectively reads that tag and then packages the protein in an appropriate vesicle and whisks it off in to its destination. That could be going back to the nucleus, that could be going to a mitochondrion, it could be going off to the cell surface in the cell membrane. But that's all happening out here in the Golgi apparatus. A final thing that's mentioned at the end there is that the Golgi apparatus also is responsible for creating lysosomes. Lysosomes aren't super important for anything we're going to be talking about. These are basically digesting organelles. So these would be basically little vesicles, so you can imagine this could be one, that contain hydrolytic enzymes that are basically just in charge of digesting materials. So a white blood cell, for example, creates lots and lots of lysosomes because white blood cells roam around, consume bacteria, viruses, other sorts of debris, and then destroy them with these lysosomes. Neurons make them too. There are some functions for them. You know, as mitochondria die within a neuron, the lysosomes will come along and chew up those mitochondria and liberate the materials for so they can be reused. But otherwise, they're not super important for what we'll be talking about. Any questions so far? Try not to go too fast, but also not to go too slow, depending on how much review this is. All right, another really important structure, another membrane-bound structure that exists within neurons as well as all other cells for the most part, are mitochondria. Mitochondria are really interesting. They actually, I mean, their main job is that these guys are, they're referred to often as the power plant of cells. These are massive ATP producers. ATP is this energy currency of cells. So we use ATP as a high energy molecule that we produce in mass, and then anytime we need to do a reaction of some kind within a cell, which is constantly the case, we have to use some ATP. And so it's the mitochondria that are responsible for making ATP. Neurons are extremely metabolically active. They have very high consumption rates of fuel, so they are producing large amounts of ATP all the time. And the reason for that is that they use ATP at a very high rate. So it's often the case that their metabolic rate is roughly 10 times that of a typical tissue. So neural tissue is extremely metabolically demanding, and that means that mitochondria are really important because they are producing all of the ATP for them, virtually all. The way it works, in a very, very concise nutshell, is that ATP will employ what's called aerobic cellular respiration, where we start with glucose molecules. Glucose molecules, through a process called glycolysis, are turned into pyruvate. And then pyruvate, this is basically a carbon-based molecule, will be broken apart into carbon dioxide. It is that same carbon dioxide that we ultimately exhale every time you breathe out. This is happening in all of our cells all the time. But as we turn it into carbon dioxide, we generate some high-energy molecules, and those high-energy molecules are used by the mitochondria to ultimately produce ATP, And the rub is they can only do so if oxygen is present. Oxygen serves as what's called a terminal electron receptor. So we use some high energy electrons, we need to get rid of them, and we deliver them to oxygen. So as long as you have oxygen, we can continue to run this process. But if you run out of oxygen, which can happen if you are not breathing fast enough relative to your consumption rate, or your breathing is somehow interrupted, then this whole process kind of backs up and slows down, and you're unable to produce ATP at an appropriate rate. But that's basically the job of mitochondria. I think they're really interesting organelles. They have their own DNA. There's very good evidence that, in fact, they produce their own genes. They're, from that DNA to make their own proteins. They have different ribosomes than are existing within the rest of the cell. There's other kinds of interesting differences. So there's very good evidence that these guys are actually derived from bacteria that were probably living within eukaryotic cells at some point, and they've sort of become part of the cell. And they've lost a lot of genes along the way, so they can't function and live on their own anymore. But they probably at one time were symbiotic bacteria that were sort of coexisting with whatever the ancestral eukaryotic cell is. I think they're really interesting. Mitochondria also, I mean, they're definitely present in abundance within the cell body because that's where a lot of the metabolic stuff is happening within neurons. But they also are transported to other parts of the neuron. So for example, they're often transported to the end of the axon. It's called the axon terminal. And so you see a lot of mitochondria down at the very ends of axons. And the reason for that is that there's a lot of ATP consumption going on there. So we'll move mitochondria around within neurons and get them to the locations they need to be in order to have efficient function of that neuron. Again, neurons are highly aerobic. They have very high ATP demand. If you have some kind of deprivation of oxygen, so you have somebody that is drowning, that can cause rapid and widespread neuron cell death. And unfortunately, neurons, along with heart cells, are probably two of the most sensitive kinds of cells to this. They just don't do well if they have to rely on any other kind of ATP production. Whereas like a skeletal muscle cell, they can switch to fermentation. They can get by for a while. Neuron cells just can't handle that. And so very rapidly you can have widespread neuronal cell death as a result of oxygen deprivation. This is another cool stat. So your brain is about 2% of your body mass, but your basal metabolic rate, so this is your metabolic rate when you're at rest, when you're not doing anything else, you're not digesting, you're just sort of, the lowest metabolic rate you can ever have at the lowest point in any point of the day. But about 20%, 1/5 of your basal metabolic rate is devoted just to running your brain, even though the brain is only 2% of your overall mass. So again, the brain is, roughly has about 10-fold higher metabolic rate than your standard tissue across your body. And interestingly, a huge chunk of that, I'm not sure the percent, maybe 40, 50% though, of that basal metabolic demand is used to run the sodium-potassium pump, which we'll talk about in the next chapter. And you've probably heard of before too. And that's all it's doing. We're just pumping sodium and potassium and burning through our energy to do that. But that sets up everything else. All right. So those are membrane-bound organelles that exist within neurons. Another really important part of neurons and any other cell is the cytoskeleton. So these aren't technically organelles, but we have this whole suite of proteins that are designed to give structure and allow function of neurons. And there's basically three major categories of these things that exist within neurons. And again, if you've had 111, bio 111, you've seen this before. But the idea is that we have, here are these three kinds. There are microtubules, microfilaments, and in neurons we call the last category neurofilaments. In any other cell we call them intermediate filaments. And there's slight differences like in terms of which specific ones that we're making in neurons versus others. But these are basically, neurofilaments are a kind of intermediate filament. They each have different jobs. So microtubules, I like to call them the superhighways of cells. So these are basically roads upon which little motor proteins will walk as they're moving around within cells. And they literally, like the little motor proteins, literally look like little sticks with feet. And so they are, I'm going to ask you to watch a video as the first part of the homework for chapter 2 that's due Monday. And that video, it's the motor proteins that really steal the show, but they all walk upon these structures called microtubules. Microtubules, you can see from this picture, are composed of lots of these little tubulin dimers. So these little pair of things here, those are tubulin proteins, and then we stick them together in this large coiled up tube, and it literally is a big, coiled up tube. And those tubes then span across the interior of neurons. And if you look in axons in particular, these have a really important role because they basically run along the length of the axon, and they little motor proteins, as I said, walk upon them and pull with them vesicles or mitochondria or other structures up and down the neurons. In fact, I think that's my next slide, so I'll show you that in a second. So they're really important in terms of that function. The next category are the microfilaments, and the main example of microfilaments that we ever talk about is actin. And actin certainly plays some interesting roles within neurons, but it's not really the star of the show. Like if you look in muscle cells, for example, actin along with myosin are really important in terms of allowing muscular contractions. You do have actin present within neurons, and actin can be involved in movement, so especially like those little movements we were saying during development, where they're growing axons and elaborating axons. Some of that is of that function is allowed by the fact that we have these microfilaments present, and so it does allow for some sort of basic cell movements. But it's not like neurons are contracting 2/3 of their length in an adult, so that they're probably less important than some other structures, at least as far as neurons are concerned. The last category are these neurofilaments, or intermediate filaments. And neurofilaments are basically in charge of producing kind of a scaffolding. So I tend to think of these as like purely structural. If you're going to have a large, large building, you have lots of supporting struts running every which way. If you look at like a skyscraper as they're building it, there's steel beams running in multiple directions to reinforce it in different angles. And that's basically what neurofilaments do. They serve and form kind of a scaffolding web around the exterior and interior of neurons that help them to hold their shape. And so certainly if there's any kind of problem in terms of the formation of these neurofilaments, that's going to play havoc on your ability to have stable structure, a stable, cellular structure within the neuron. This is another picture that kind of shows all these at the same time. So there are the little actin filaments that I'm just going to sort of gloss over for now. Here are these nice big green microtubules. I don't know why they keep showing them in green, but that's what they do. But they're kind of running in the course along the length of axons, as well as in and out of, from the nucleus out to the cell surface within the cell body. And then this crisscrossing scaffolding array, that's all these neurofilaments or intermediate filaments that are providing stability and structure. Any questions so far? All right, so I want to try something. I want to try doing a little Kahoot quiz. Have you guys used Kahoot anywhere else? A few people have. Good. I think it's kind of fun. Right. Danielle still in the lead. Question 3. Which type of stain is used to visualize the neurites of a neuron? All right. It is, in fact, the Golgi stain. So Golgi stain is the one that allows you to visualize dendrites and axons really nice and clearly. The Nissl stain is what is very reliable in terms of finding all the cell bodies and such. And sometimes you can kind of see the axons, but it's pretty tough. The Golgi stain is what does it. Horseradish peroxidase, we'll talk about, is really cool. It allows you to figure out if you know where a neuron ends, or if you're interested in where a neuron's axon terminal is, you can inject there and it will take it up into the rest of the cell. We haven't talked about it yet though, and that's why nobody marked it. And Gram stains are used in bacteria. All right, SL. Good job, SL. Question 4. Which of these functions as a superhighway for motor proteins and neurons? It is, in fact, the microtubule. So microfilaments are things like actin that allow for some movement, but they're not really allowing motor proteins to move along them. All right, awesome. So, oh, Sam took the lead at the very last second. SL is microseconds behind. Okay, I think these are kind of fun. I will try to include them whenever I can. They're just meant to be kind of a checkup to see how we're doing if you're processing and understanding everything. All right, I got 2 minutes, so I'm going to take 2 more minutes. In fact, actually, maybe I won't. Let me just set this up. So what we've done at this point now is we've done an overview of most cell parts for a general cell. What I want to do now, and starting, we'll do this on beginning of class Friday, is turn our attention to neurons specifically and talk about specific structures that we see in terms of things inside axons and dendrites and how they work. And we'll talk about classification of neurons. And then from there, we'll have time to work on the homework, I think. So please do bring and be prepared to have some time to work on homework on Friday. Probably be the only time this semester we do that, but that's okay.